Flow cytometry is an analytical technique that quantifies the frequencies of cells binding to fluorescent antibodies and scattering light in characteristic ways. Flow cytometers use laser beams to provide fluorescence excitation and a series of detectors to identify both scattered light and fluorescent signals from single cells flowing in a focused stream past the laser. The simplest cytometers use a single argon laser emitting a high-intensity beam. But more complex cytometers use lasers delivering 8 or more distinct wavelengths. Let's examine the steps of flow cytometry. First, a heterogeneous population of cells in a well-mixed suspension is introduced to the instrument at the sample injection port, or SIP. Within the SIP tube, the suspension is mixed with a saline sheath fluid which encircles the narrow stream of cells, hydrodynamically focusing the sample and ensuring that cells pass single file through the laser beam. As each cell passes through the beam, most of the light is scattered forward. The amount of forward light scatter provides a rough measure of the size of the scattering cell. The more forward light scatter, the larger the cell. This light is detected and recorded by a photodiode. A second light scattering detector is located at approximately 90 degrees to the path of the laser beam. It picks up the amount of side-scattered light, which offers an indication of the degree of intracellular complexity of the scattering cell. The more intracellular membranous structures present in the scattering cell, the higher the level of side scatter. Since much less light is detected by side-angle scatter detectors than by forward scatter photodiode detectors, side-scattered light is measured by more sensitive detectors, called photomultiplier tubes, or PMTs. PMTs convert light energy into electrical energy. Each cell passing through the beam generates a pulse of electrons within any PMT that receives the light signal from it. The pulse of electrons is translated to a voltage pulse, which is measured, digitized, and recorded by the instrument. The wavelength of the scattered light remains unchanged from that of the incident beam. However, in addition, some of the incident light interacts with fluorescent dyes bound to surface antigens on particular cells and is re-emitted as fluorescent signals at a wavelength characteristic of each fluorescent dye. The fluorescent light coming from labeled cells moves through a series of filters and mirrors so that specific wavelength ranges are delivered to the appropriate detectors. The flow cytometer counts every cell as it passes through the laser beam and records the level of emitted fluorescence at each specified wavelength, as well as the amounts of forward and side-scattered light for each cell. The information is integrated by the cytometer software and can be expressed in a number of formats, such as histogram, contour plots, and scatter plots. Thus, flow cytometry enables scientists to gather key statistical data on large numbers of cells and use that information to correlate multiple parameters within a population.